Back. Today we are taking up nucleotide metabolism part 1, that is purine synthesis. Before we really begin, I would like to chant this Shanti part from Dietary Upanishad. This is a very common chant. You might have all heard about it. Om Sahana Vavatu Sahano Bhunaktu Sahaviryam Karavavahai Tejasvina Badhitamastu Ma Vidvishavahai. Om Shanti Shanti Shanti. Before beginning a session or ending even one, we can chant this. The meaning is, it's a one-to-one relation. It is Sahana Vavatu, means it's a two-person conversation, that is between me and the student. Even though at one point of time 100 of you will be listening, but I am teaching only to you and you are listening only to me. So I have one-to-one relation to all of my students, those are attending this lecture. Similarly, it is invocation that we should learn together, let the God protect we two together, let us have knowledge together, let us have courage, and we should not have any conflicts, we should not have any doubts before leaving the session. So such a great message, and again this Shanti 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 to create real peace of mind to learn things. This lockdown has taken us 5 steps up, making us all aware of these online classes, video lectures and what not. You might be munching your 5-star bar or might be having a bowl of snacks beside you, chewing gums, but I would suggest that you focus your mind on the video lecture that you are attending and think that as if you are listening to me in the lecture hall. So coming to the nucleotide metabolism, why we should be actually bothering about it. It's a very amazingly intricate, highly regulated enzymatic machinery that regulates many processes, particularly if we are knowing about purine and pyrimidine synthesis. That means we are knowing how the DNA and RNA amount can be regulated in our body. So that is all about it. We need to kill some microbes, so we need antibiotics. We need to reduce their purine and pyrimidine synthesis so that their DNA and RNA can be synthesized properly and their growth slows down. Or another category of groups drugs, we want to kill the human cells by reducing purine pyrimidine so that the cancer cells don't grow much, and we add up some other drug to kill the cancer cells as well. Apart from this, we have certain diseases related to this purine and pyrimidine synthesis like Lesch-Nyhan syndrome, SCID or bubble baby syndrome, so which are not very common in India, but gout, that is very common in Indian context. So all this will be dealing in this class. Now where from we get nucleic acids? They are available everywhere. In all the food products they will be there. So wherever there is growth, there are DNA and RNA involved. So we get from all plant products, but they are rich wherever there are muscles. Since muscles are rich in ATP, we get lot of nucleotides from that. Yeast, it's a natural source of RNA, and for laboratories also it's a good source of nucleotides. But it is not the dietary nucleotides that actually make the nucleic acids. So actually these are not essential for us. Even if we do not take them from outside, our body has its own machinery to synthesize them. So, both purine and pyrimidine can be synthesized from different amphibolic intermediates, but these dietary nucleotides do enter and they are handled in our body. So, what happens to them? Once we ingest dietary nucleic acids, they are dissociated and free DNA and RNA are obtained. So, what are DNA and RNA? They are the nucleic acids. If you remember, nucleic acids, nucleotides, nucleosides and nitrogenous base. Nitrogenous base plus sugar is nucleoside, nucleoside plus phosphate nucleotide, and these nucleotides along with sugar and phosphate, this backbone, they form the DNA or RNA, along with that some proteins also. Now, this DNA and RNA, they are worked upon by either ribonucleases or deoxyribonuclease as per the nucleic acid, and also polynucleotidases. They break down and form mononucleotides, and they will be acted upon by nucleotidases, phosphatases, which will liberate the nucleosides. Now, these nucleosides will be acted upon by phosphorylase. I had told you they contain now only sugar and nitrogenous base. So, that will liberate the base and deoxyribose or ribose sugar. Now, they will be oxidized and excreted. So, the sugar may be takes its own course. What happens to the bases? They are not all excreted by oxidation, rather they get repeatedly used, that is called the salvage pathway. So, what are those? That we will see little later. If you see the overview of nucleotide metabolism, the beginning product is PRPP, that is phosphoribosyl pyrophosphate. The pyrimidine nucleotides after synthesis, how they will be dealt with? They will release the beta alanine along with some other products also, and purine nucleotides, they will release uric acid. It is a toxic waste product, but no, it is not all bad. It has also got some antioxidant properties that we will see later. So, beginning product is PRPP, pyrimidine and purine nucleotides both are synthesized by that. Pyrimidine nucleotides catabolize to beta alanine and some useful metabolites which are reutilized. Purine nucleotides gives rise to uric acid. Remember this. Next we will proceed to the biosynthesis of purine nucleotides, which is today's lecture. So, most of the cells, they will synthesize this. The major organ is liver and it takes place in cytoplasm. Two pathways are involved, that is de novo pathway, new synthesis from amphibolic intermediates, or the salvage pathway. It is from either the intermediary catabolic products of the purine catabolism. So, free purine bases either will be rephosphorylated or purine nucleosides will be phosphorylated to give rise to nucleotides. Fine. Coming to the first pathway, that is de novo pathway. Now, what does this de novo means? De novo means newly synthesized purines. So, as I told you, the beginning product is PRPP. After some 11 steps, the first purine nucleotide is obtained, that is IMP, first purine nucleotide, inosine monophosphate. So, in one pathway we obtain AMP, that is adenosine monophosphate, from their ADP and ATP by AMP kinase and nucleoside diphosphate kinase respectively, and in another pathway we obtain GTP. So, this is the overview of the first pathway of purine synthesis, de novo pathway. Fine. Now, this purine ring, as we have seen, it is comprises of two rings. It is not from a single source. Different products, they donate the carbon and nitrogen atoms, like the N1 is obtained from aspartate, C4, 5 and N7 they are obtained from glycine, and from glutamine amide nitrogen, 2 nitrogen atoms of N3 and N9 are obtained. Rest 2 carbon are donated by to 1 carbon moiety, that is N10 formyl tetrahydrofolate and N5 N10 methynyl tetrahydrofolate. So, in nutshell, we get these 2 nitrogen atoms, the 4 nitrogen atoms from 3 amino acids, aspartate, glycine, glutamine. The C6 is obtained from respiratory carbon dioxide, and 2 1 carbon moieties, they provide the C2 and C8. Okay. So, protein gives rise to nucleic acids in one way, if we think so. Wow. So, is this purine synthesis? You might be screaming in pain. Am I going to remember all this? Oh my God, wait, we are not going to pounce upon it like that. Let us see the first step, formation of PRPP, that is phosphoribosyl pyrophosphate. So, the beauty is the sugar, that is alpha D ribose, that takes up one after another product, and gradually the ring builds up. The first step is in presence of PRPP synthase, 1 ATP is utilized, but here 2 activated phosphates are donated to the sugar. So, these 2 phosphate atoms together, they are called as pyrophosphate, and thus it is phosphoribosyl pyrophosphate. Phospho for the phosphate groups, ribosyl for the ribose sugar, and pyrophosphate because it is PP. Okay. So, now we have got our beginning product. So, this is common for both the purine and pyrimidine. Now that we have arrived at PRPP, this PRPP is acted upon by PRPP glutamyl amidotransferase. So, what the name suggests? PRPP glutamyl amidotransferase, that is from glutamine molecule the amido group is being transferred onto PRPP. So, what it becomes? It becomes 5-phospho beta deribosylamine. So, the amino group has been transferred from glutamine, hence glutamine becomes glutamate, and then next step is another ATP is utilized and there is some rearrangement and we get glycinamide ribosyl 5-phosphate. What we got? GAR, glycinamide ribosyl 5-phosphate. Then in presence of formyl transferase and this N5 N10 methynyl tetrahydrofolate, there is transfer of this methynyl group and we get formyl glycinamide ribosyl 5-phosphate. So, a big name. The next step is the 5th step. In presence of synthase, again another glutamine will donate its amino group, and what we get now? Formyl glycinamidine ribosyl 5-phosphate. So, there might be little bit of nomenclature difference in different books, and after this product is formed in 6th step, in presence of ATP and magnesium, there will be ring closure with removal of one water molecule. What we obtain is aminoimidazole ribosyl 5-phosphate. Then the respiratory carbon dioxide, what it does? The 6th carbon atom it will donate. So, in presence of carboxylase, it will produce aminoimidazole carboxylate ribosyl 5-phosphate. So, we call in short ICAR. Now, in presence of synthetase enzyme, aspartate will get attached and water will be removed to form aminoimidazole succinyl carboxamide ribosyl 5-phosphate. In short, it is also termed as SICAR. So, the previous one was ICAR, this is SICAR. From SICAR, this fumarate is removed in presence of adenylosuccinase, and what we obtain is aminoimidazole carboxamide ribosyl 5-phosphate. Next step, formyl transferase, another 1 carbon moiety N10 formyl tetrahydrofolate will donate the formyl group and it will finally produce formimidazole carboxamide ribosyl 5-phosphate. So, till now the ring is open, and now in presence of IMP cyclohydrolase, as the name suggests, cyclohydrolase, hydrolysis occurs, so water molecule removed and cycling, that is the cycle is formed, the ring is formed and inosine monophosphate is obtained. So, as we are discussed, from PRPP, the first nucleotide that is being formed, ribonucleotide, after 11 steps we are getting inosine monophosphate. From here now there will be bifurcation. In one pathway there will be adenosine formation or ATP formation, in other there will be GTP formation. So, adenine and guanine, they are the purines, right. Now, this IMP conversion to AMP and GMP, see the beauty, GTP helps in formation of ATP and ATP helps in the formation of GTP. Now that we have reached IMP, now another aspartate comes and in presence of adenylosuccinate synthase, it gives rise to adenylosuccinate, that is AMPS. This again is acted upon by adenylosuccinase and we obtain adenosine monophosphate. Similarly, the other pathway we will see from IMP. In presence of IMP, dehydrogenase. Dehydrogenase here NAD plus will produce NADH plus H plus, that is 2 hydrogen atoms are removed in presence of water and we get xanthosine monophosphate. So, xanthosine monophosphate will finally in presence of transamidinase and glutamine. Glutamine will transfer its amino group and guanosine monophosphate will be obtained, that is GMP. Fine. So, now we have got AMP and GMP. So, after inosine monophosphate in GMP we are getting xanthosine monophosphate. So, when you see XMP, do not get confused, immediately think about guanosine monophosphate GMP. So, this can be a very good quiz question for you. Then regulatory control of purine biosynthesis. How it is controlled? If it goes unchecked means we land in excessive of the nucleic acid synthesis, again that will lead to excesses of catabolism and uric acid may increase. So, one thing is concentration of PRPP and feedback regulation at different sites, they will control the purine biosynthesis. Now, coming to the first thing, that is our most important product, that is PRPP. If it is more, then purine nucleotides are also largely synthesized, and PRPP synthesis depends upon ribose 5-phosphate presence and on the activity of PRPP synthase also. Now, if you recall, if there is more of HMP shunt, because ribose is obtained from HMP shunt. So, HMP shunt is more active means it will channelize to produce more of ribose 5-phosphate. More ribose 5-phosphate means PRPP will be activated and more purine synthesis. Then very importantly, the feedback regulation. PRPP synthase can be feedback regulated or inhibited by purine nucleotides like AMP, GMP and IMP. All the products that are formed, they can feedback inhibit. So, now yes, I am enough synthesized, you please stop. The committed step, the second step from PRPP to phosphoribosylamine, again a quiz question for you. So, PRPP glutamyl aminotransferase, that is the enzyme, it is also inhibited by AMP and GMP. Now, AMP feedback regulates adenylosuccinate synthase and GMP feedback regulates IMP dehydrogenase. So, ATP and GTP they also cross regulate. So, these are the 4 ways how the feedback regulation acts. Repeating again, PRPP synthase is feedback inhibited by purine nucleotides, the end products AMP, GMP and IMP, the intermediary products as well. Then the committed step enzyme, the second step, PRPP glutamyl aminotransferase enzyme is inhibited by AMP and GMP also. AMP feedback regulates adenylosuccinate synthase and GMP feedback regulates IMP dehydrogenase. So, the intermediary enzymes are being checked by AMP, GMP also. Apart from that, ATP and GTP, ATP enhances GTP synthesis, GTP enhances ATP synthesis. So, that way they cross regulate each other. Now, let us see the overview, control of de novo synthesis. As I had told you, ribose 5-phosphate to PRPP, after that 11 steps, and we are getting IMP in between 5-phosphoribosylame, that is the committed step also here being mentioned, and you can see how AMP and GMP, even XMP, all are feedback regulating the initial enzymes. So, GTP is cross regulating AMP synthesis, ATP is activating GTP synthesis. So, all these are the different ways how purine synthesis is kept under check. Now, by understanding the process, the entire synthesis process, and where the regulatory steps are working, we can design our drugs. So, particularly antifolate drugs and glutamine analogues, they affect the purine synthesis. Antifolate, how? Because we have two 1 carbon moieties like the tetrahydrofolates. If they are checked, then there will be control of purine synthesis, and certain glutamine analogues also, they can act upon different steps. Now, coming to the carbon of this tetrahydrofolate, the methotrexate, the folate antagonist, the folate antagonist like methotrexate, they diminish this tetrahydro 1 carbon moieties, and they are used as anti-cancer drugs, as I had told you. Likewise, azathioprine is a glutamine antagonist, impedes the step 2 and 5. Diazo-norleucine affects the step 2. Similarly, 6-mercaptopurine, it affects the step 13 and 14. Conversion of IMP to AMP or GMP, they are all affected. Then mycophenolic acid reversibly inhibits IMP dehydrogenase, that is the step 14 is impeded. Now, we will discuss little bit